Okay, so today we're going to talk about movement of molecules and how does a cell get the materials it needs to function. So, like we talked about before, our cells have these semi-permeable membranes, and cells need to be able to move materials in and out of their cells through their membranes. So let's talk a little bit more specifically. What does semi-permeable mean? So when we talk about semi-permeable, we're looking at a type of membrane that allows some things to move across and some that cannot. So you'll notice that on the left, these fat-soluble molecules are able to move freely, and then these water-soluble molecules move through a channel protein. So this is able to control what's coming in and out of the cell through these actual channels, while other things can move freely. When we talk about fat-soluble and water-soluble, we don't actually mean water and fat molecules, just things that are soluble within that, which we'll talk more about later this year. So semi-permeable means that some molecules can move across it while others cannot. And there's two different methods that we're going to talk about. And there is another method, but today we're going to talk about diffusion and we're going to talk about osmosis. And in both of these different methods, molecules are moving from an area of high concentration, where there's a lot of those molecules of that specific type of molecule, to a low concentration, where there's a lower amount of that specific type of molecule. So when we talk about diffusion, we're talking about solutes, and they're moving from high to low. So you'll notice in this you have these small molecules in this extracellular fluid, and extracellular just means the fluid outside of the cell. And then you have this plasma membrane, or this semi-permeable membrane. And over time, which goes across the bottom, you'll see that these nonpolar molecules are slowly moving across the membrane into the cytoplasm of the cell until they reach an area of equilibrium where there's the same amount of molecules on each side. Once this occurs, molecules will still move back and forth, but it will be in equilibrium, so if one moves over, another one will move back, and so that both sides stay about the same. When we're talking about osmosis, we're specifically talking about the concentration of water. So this also moves from high concentration to low concentration, and it flows freely across the membrane. So if we have this beaker set up with a semi-permeable membrane, and one side has a lot of salt molecules, it means there's not a lot of free water molecules not bound to the salt in that side. So what you'll notice, in order to create this equal concentration of water, is water will leave the left side and go into the right side, pushing that water level up, and this can actually be done in a beaker or another type of apparatus that will help create this type of simulation, where you'll notice that one side is higher than the other because of the salt molecules. And the salt molecules are not able to move back and forth, which is why the water moves over, so that the amount of free water, or water not bound to the molecules, remains equal on both sides. So if you were to look at this experiment right here, this is a dialysis tubing experiment, which means that membrane, or that dialysis membrane, is actually permeable to some things and not to others. So inside at the start, you have this really concentrated solution within the bag. And what you'll notice is that the blue molecules, sorry guys, could move. The blue molecules cannot move freely. That's why they all stay inside, because that membrane is not permeable and does not let the blue molecules move through. This might be because they're larger and it might only let smaller molecules move. But you'll notice that the red molecules were able to move outside of the bag to create this level of equilibrium, so that for the surface area, there's the same amount inside as there is outside. So all molecules are moving towards this equilibrium. If you were to actually set up this experiment in the lab and put starch inside of the bag, inside of a water, starch and glucose inside of a bag, inside of a water beaker, if you were to then leave it sitting there, if after 10 minutes you tested the water on the outside, you would notice that it does test positive for glucose because it's a smaller molecule, but it wouldn't test positive for starch because the starch cannot move through and would stay inside of the bag. When you're done, make sure you go do your worksheet and then work on your TED Talk section. I believe for today, your worksheet goes along with an online lab, and the link is on the website.